It's just about 12 noon, so we'll get started. Thank you everyone for joining us for the first webinar of the Clinical Trials Methodology course, the webinar series for 2017. My name is Sam Puliso-Busterian. I am the project manager for the CTMC at the University of Michigan. And our webinar topic today is asking a good early phase clinical trial question presented by Dr. Roger Lewis. So just a few housekeeping items before we get started. Everyone but our speaker today will be muted. This is to prevent feedback during the presentation. But we do encourage questions, so please use the chat box to type questions for Dr. Lewis. If you are interested in CMEs, you must complete an evaluation form, and we will provide details at the end of the webinar. So it's my great pleasure for now to introduce our distinguished speaker. Dr. Roger Lewis is the chair of the Department of Emergency Medicine at Harbor-UCLA Medical Center. His academic interests center on adaptive and Bayesian clinical trials, translational health services, outcomes research, interim data analyses, the role and function of data monitoring committees, and difficulties surrounding informed consent in emergency research studies. Dr. Lewis has served as a grant reviewer for numerous national and international institutes and foundations, including the Canadian Institutes of Health Research, the National Cancer Institute of France, the CDC, the NIH, the CORI, and many others. He also has served as the chair of DSMBs for both federally funded and industry-sponsored clinical trials, and he is a research methodology reviewer for JAMA and is past president of SAEM. Dr. Lewis is a senior medical scientist at Berry Consultants, a group that specializes in adaptive clinical trials, and he's also a member of the executive committee of our course. So Dr. Lewis, thank you so much for joining us today.
Great, thank you very much. It's a pleasure to be here. Welcome to everybody. I will try to talk and keep an eye on the chat box at the same time, but Sam, if I am missing something, please jump in. So today what we're going to talk about is the goals and their associated questions that should be asked and hopefully answered by an early phase clinical trial. And my goal here is my goal in the presentation, I should say, is to put the context of an early phase clinical trial in light, or to define the context in light of the overall plan or the project in terms of demonstrating the efficacy of a device or a drug or even a diagnostic strategy, so that you think of an early phase clinical trial as a step in a much longer project or process of identifying new effective therapies, treatment strategies, or diagnostic strategies for patients with neurologic diseases. I have a number of financial disclosures. I'm lucky enough to get support from a number of sources, and I work very closely with Berry Consultants, and they have multiple clients, many of which are doing work in this area. In addition, I'm going to talk about a particular clinical trial that's NIH funded, but involves the investigational use of L-carnitine, and I just need to make it clear that that agent is not approved for the treatment for which it is being studied. So what is an early phase trial? In order to understand what an early phase trial is and is not, it's important to realize that for us to demonstrate ultimately that a treatment is adequately effective and safe so that it warrants clinical adoption, we must be able to define the right population in which that treatment ought to be administered, the target disease or diseases, and those diseases may be characterized both by the general diagnosis or by severity or stage, the treatment strategy. How do we actually apply that therapy? What is the timing, the dose of the medication, the route by which it should be administered? We need to understand the outcome that we are trying to affect. For some diseases, the outcome may or may not be of interest to particular patients, and that may influence whether or not the treatment is warranted. And we need to be able to define a confirmatory trial design that will evaluate the use of that treatment in that population for that disease using that treatment strategy in order to demonstrate a positive effect on the clinically relevant outcome of interest. Early phase clinical trials are designed to reduce uncertainty in each of these areas, the population, the disease, the treatment strategy, and even the outcome, so that we are able to run a successful confirmatory trial. A successful confirmatory trial is not necessarily one that gives you a positive result. It is one that tells you unambiguously whether the treatment is of clinical value in the target population. So we often think about the phases in the development of a drug, but there's an associated series of steps in the development of a device as well. There's an early stage, phase 1 in drugs, and sometimes this is bench work in devices, where we're trying to learn very basic things about the treatment that we are investigating. Then we may analyze the results of that work, we make a number of decisions, and we enter phase 2. In phase 2, we leverage what's been learned in the earlier work. For example, the maximum tolerated dose, or the side effect profile, or the pharmacokinetics. And we start to look at outcome results. In after bench work for a device, we may do some early clinical testing to make sure the device can actually function when implanted as it is intended. We take the results of those early stage work and we make a number of decisions regarding how to design a confirmatory trial, usually called a phase 3 trial in the setting of drugs. But in a device trial, it's often called a pivotal trial, to verify the preliminary evidence of efficacy we've obtained earlier and just demonstrate unequivocally the effectiveness of the treatment to improve the outcome. But this leads to sort of a general concept, there are some stages in development, which we call the learn stage, and then there are some later stages in development where we hope to confirm or verify what we already believe to be true, or at least hope to be true. In the drug development, the learn phase is considered phase 1 and phase 2, and at that stage, we are really trying to answer a variety of questions, and so there must be greater flexibility in the number of questions that we ask and the goals. We may be interested in toxicity, feasibility of administration, side effect profiles, variations in pharmacokinetics across populations, etc. Because we are addressing multiple questions and learning in a more flexible way during the learn phase of investigations, we have to have a willingness to tolerate higher error rates, and there may be some sources of bias. For example, we may have a highly selected patient population, for example, one that has failed prior treatments, and that may lead to bias in the estimate of a treatment effect or side effect profile relative to the effect size or the side effect profile we would see in an unselected population. But it is important to understand that phase 1 or especially phase 2 trials are not just small underpowered confirmatory trials. They actually have different goals. In contrast, during the confirm phase of investigation, so this would be termed a pivotal trial in device work or a phase 3 trial in drug work, we want to have rigid control of error rates. So we'll have a very rigid control of our type 1 or false positive error rate, a defined power, meaning the probability of getting a positive result if the treatment has a specific and pre-specified treatment benefit. We're going to pre-specify and be held to that pre-specification in defining a single primary question or outcome that defines whether that phase 3 trial is positive or negative. So this is the setting in which we often use a traditional one-to-one randomized controlled trial. And I don't know why randomized was used twice there, but that's my slide. Okay. So I'm going to now go through a series of early phase questions and talk about the designs that are commonly used in early phase work to answer those questions. In many cases, there are multiple questions, and we might work to develop a trial design that is an amalgamation or a combining of these different approaches. So the first question that's often asked is, can the drug or the device be tolerated? In the case of a drug, this is a traditional dose escalation type question, and it may be conducted in a healthy population or a diseased population. If you're asking, if you're trying to assess the maximum tolerated dose in a healthy population, it's always important to assess whether or not a diseased population can in fact tolerate the same dose or has the same pharmacokinetics or pharmacodynamics. I'll stop for a second and point out pharmacokinetics is the term used to denote things like the serum concentration over time. Pharmacokinetics is the term that addresses the actual effect, the pharmacodynamics is the actual effect of the drug over time, which may not exactly mirror the drug levels or the pharmacokinetics. Typical designs of clinical trials that are used to assess maximum tolerated doses are 3+3 designs. These are designs in which we give 3 patients a low dose, see whether or not anybody has a dose-limiting toxicity, and then if not, we try a higher dose in the next 3. And then there are rules that are pre-specified that tell us when we've decided we've identified the maximum tolerated dose. This design is inefficient, it's often inaccurate, and it's primarily of historical interest, but one will still see proposals that include that as the proposed trial design. It was often used, for example, to identify the maximum tolerated dose for oncology studies for chemotherapeutic agents. A better approach is something called the continual reassessment method, which is an approach in which there's a mathematical model for the relationship between the dose that's administered and the probability of a dose-limiting toxicity, and that distribution function or mathematical relationship is updated with all new data, with each new data that comes in, each patient experience. And then that overall relationship that becomes estimated more precisely over time with additional data is used to decide what dose is given to the next patient. This is a more efficient and more accurate method for doing dose ranging or dose finding, especially for toxicity. A second early phase question is the question of whether we can establish proof of concept. Proof of concept refers to this idea that we often have a preconceived theoretical notion about how the drug or device ought to work. So, for example, if the drug is intended to engage a particular target, then demonstrating that that drug, when administered in a human, actually binds to the target receptor might be considered one step in proof of concept. A later stage in proof of concept may be to show that when it engages that target, it either blocks other messengers engaging that target, or for example, triggers intracellular or extracellular processes that the target is thought to mediate. We may want to know whether the drug actually passes the blood-brain barrier if the target is in the CNS. And there are a number of somewhat embarrassing situations in which clinical trials have been run only to find later that the drug never actually got to the location of the disease in question. The assumption when asking a proof of concept question is that the treatment strategy will not work without this criterion being met. So if the tumor is in the CNS and the drug cannot get to the location of the tumor, we are willing to conclude that the drug is unable to be effective and therefore further testing is not warranted. The trial designs used to address the proof of concept include dose ranging studies, biomarker studies, or assay studies, often looking at where the drug is getting to and whether it's engaging the target that is its primary presumed mode of effect. The next early phase question is often what dose should be investigated further, meaning what dose or dosing regimen of the drug is worth further investigation. And early on in the testing of a new investigational agent, the potential doses that may be of clinical importance may span several logs. It is not uncommon to have initial dose information be based on rodent models or other non-human models. And experience has shown that those types of models are highly inaccurate in identifying the doses that will be either effective or successfully tolerated in humans. Dose selection often requires a balance of efficacy and toxicity. It's always possible to find a dose with no toxicity, that's 0. But finding a dose that is likely to succeed in confirmatory testing requires finding one whose toxicity will not be so dose limiting or treatment limiting that patients can't tolerate the treatment, but high enough so that the drug can have the intended effect and demonstrate efficacy. It is a very common mistake in drug development programs, even those run by very large pharmaceutical companies, to narrow the dose range under consideration too early. For example, to use very sparse data obtained in early phase trials and make a best guess at the dose, only to find that in confirmatory testing the dose either couldn't be tolerated, or in fact more commonly had inadequate efficacy, raising the question that in fact the dose that was selected for further study was too low. So the trial designs used to address this kind of question are dose-finding trials, sometimes called dose-ranging studies, and they are best conducted with measures of both efficacy and toxicity. And I will give you an example of an adaptive dose-finding trial to illustrate some of the considerations. The next early phase question is what outcomes should be measured. We want to measure outcomes that are both likely to be positively impacted by the treatments we are investigating, but also have clear clinical importance and are patient-centered. We need to consider practical issues such as our ability to measure the outcome. We may be most interested in the cognitive level of a patient 10 years from now, but the timing of that would make that measurement virtually impossible to use in an early phase clinical trial. We also need to assess the difficulty in assessment. For example, the difference between doing a modified Rankin scale over the phone or in a single visit versus very detailed neuropsychiatric testing that may require multiple visits over time and more specialized training on the part of the person making the assessment. We need to consider reliability of outcome measures, which is the characteristic of how closely repeated applications of the same outcome measure will give you the same result. And we need to consider validation, which is the concept of how closely the outcome measure measures the underlying characteristic of the patient that we're most interested in. So it is important to remember that a car with a dead battery is highly reliable. It gives you the same result every time, but it's not valid as a means of transportation. We need outcome measures that are both reliable and valid. It is impossible to be valid without being reliable, but it is absolutely possible to reliably get the wrong answer if you use the wrong outcome measure. We can consider in early phase clinical work multiple outcomes. For example, we may have multiple measures of the disability associated with a neurologic disease with the idea that we are going to pick for further use in later clinical trials the outcome measure that appears to be most responsive to the treatment that we are investigating. When we do that, however, we need to consider the possibility of cherry picking. Cherry picking is a term we often use for the error that occurs when you're considering multiple conclusions or multiple measures. You pick the one that's most positive without considering the chance that the differences between them may have occurred by chance. So, for example, if you have multiple measures of functional disability after stroke, all of which measure roughly similar or related characteristics, the outcome measure that is most responsive to a therapy in one trial may not be the one that just turns out to be most responsive in a second trial of the same intervention. So we need to sometimes take statistical steps to minimize the risk of a false choice due to the cherry picking and the random variation among outcomes. The trial designs that we use in this setting include the intervention, and we may consider simultaneously measuring multiple outcomes to assess their validity, reliability, ease of measurement, and patient-centeredness. The next question is what disease state should be studied. It is often assumed that we choose therapies based on a particular disease, but in many types of clinical research, we may have a therapy that has the potential to positively impact multiple different diseases or different diseases that are related or subtypes of the same disease. When we're considering what disease states should be studied, we should consider the availability of subjects, the confounding factors and other treatments that may be administered, and the outcome measures that have been established for those disease states. It is common in early phase clinical work to enroll patients with very narrowly defined disease states and then be surprised when the treatment behaves differently in a confirmatory study when we expand to a broader inclusion a set of inclusion criteria and a broader population. We should, in general, try to run our phase 2 trials in populations as similar to the population we intend to use in our phase 3 trials, so our phase 2 trials can be most informative regarding the phase 3 design. It is common in drug development, especially, to be running simultaneously multiple phase 2 trials of the same agent in different diseases. There is some inefficiency to doing that in separate clinical trials, so we often consider clinical trials in which we're going to enroll multiple patients with related but different diseases or disease states or severity into the same trial, knowing that we may select only one of those populations or subpopulations for further testing in a confirmatory trial. So sometimes we do something called an indication finding trial when we enroll these multiple disease types into the same trial, and that allows us to obtain some efficiencies in terms of leveraging our investment in the sites and the personnel. This requires a single statistical approach to analyzing the data as a whole, however. The next early phase question is what population should be enrolled. We consider sometimes narrow populations versus broad populations, and there are arguments in favor and against both. For a narrow population, we will have less variability in the disease state, and that makes it easier to interpret the results. However, when you have very narrow enrollment criteria defining a narrow population, it is difficult to enroll your required sample size, and the results that you get are less clinically relevant since our clinical practices are characterized by broad populations. When instead we enroll a broad population in a clinical trial, there is increased variability. For example, the disease trajectories may be different over time. There may be confounding factors, for example, a variety of concomitant treatments that are received in routine clinical care. And this raises a concern regarding a very important concept, which is the heterogeneity of the treatment effect. Heterogeneity of treatment effect refers to the possibility that the actual magnitude of the benefit associated with your treatment will vary based on patient populations. This is the same thing statistically as an interaction between the treatment effect and whatever the variable is that defines the patient population's membership in each of the patient populations. Clinical trials often have very low power to detect, meaning it's very unlikely you'll be able to tell when there is heterogeneity of treatment effect. So you will not know that your treatment works better in patients with the more severe disease or the less severe disease or the earlier age of onset, etc. And therefore, when we enroll broad populations, we need to think carefully regarding the likelihood that the treatment benefit will vary by subgroup within the population and how we're going to handle that both in the analysis of the early phase clinical trial and in the decisions that we make moving forward into later phase testing. But as I stated earlier, in general, we want the learn phase trials to most directly inform the design of confirmatory phase trials and tell us whether we should be running a confirmatory phase trial at all. So we need to enroll patient populations in our early phase work that closely resemble the populations that we intend to enroll in our later phase work. The next early phase question is, how should a confirmatory study be designed, implemented, and analyzed? As I mentioned, we want our learn phase trials to be conducted in similar settings with similar populations so that they can help us answer these questions. We want to use the same populations, treatments, and outcome measures whenever possible. And we need our early phase work to enroll enough patients so that they actually answer our questions regarding the confirmatory trial. I mentioned earlier a common error in drug development in which inadequate amount of information is obtained in your phase 1 and 2 work to make an informed decision regarding dose. And people often are in such a hurry to get to phase 3 that they just pick a dose that they think is best based on inadequate information, a decision that they learn to regret later when the dose is either overly toxic or inadequately effective. The motivation for this error, the underlying issue, is that people are often in a hurry to get to phase 3, and they forget sometimes that the goal here is to be successful in phase 3, not just to get there first. The next early phase question is, should a confirmatory study be conducted at all? We should only run confirmatory trials which are inherently large, difficult, and expensive, and frankly expose large number of patients to treatments that may ultimately shown to be ineffective, whether it's the control arm or the experimental, if those confirmatory trials have a substantial chance of showing benefit. And a key concept that I'd like to introduce here, and I'd like you to think about all through the course, is that of predictive power. I mentioned earlier that the power of a clinical trial is the probability that it will give you a positive result if the treatment has exactly the benefit that you define in your alternative hypothesis. So the power that we use in designing a clinical trial has very little to do with the probability that the trial will give you a positive answer in real life. And that's because the treatments that we test rarely turn out to have exactly the efficacy that we hoped for at the time that we launched that confirmatory trial. So predictive power is a different concept. It is the probability that you get a positive confirmatory trial considering all of the remaining uncertainties in the true amount of benefit. So during your phase 2 work, you will often have administered your treatment of interest and measured your outcome of interest, and ideally your phase 2 work should be randomized and include a control group. The relatively smaller data sets you get in phase 2 work will reduce your uncertainty in the magnitude of the true treatment effect, but there will still be tremendous uncertainty. The predictive power of your confirmatory trial is the probability that you get a positive result which is obtained by averaging over all of the uncertainty in the true treatment benefit. Now, sometimes people in some neurologic areas run trials that they know have extraordinarily low probabilities of being successful, because if the trial were successful and demonstrated a real benefit, the positive effect on humanity would be so large. So an example of this is the tests of disease-modifying agents to prevent Alzheimer's disease, where the burden of disease in society is so great that if we were able to demonstrate an agent was effective in preventing or slowing progression, this would be a tremendous benefit. The challenge here, of course, is that if you run these trials with a 5% chance of a false positive result if there's no effect whatsoever, and you run 20 or 30 of them, you're likely to see a false positive. Amazingly, there have been over 30 phase 3 trials of disease-modifying agents and strategies in Alzheimer's disease without a single positive result. So we're doing even worse than you'd expect by chance. The goal here is to kill as few people as possible in the development of drug development or device development. And this requires being honest with ourselves regarding the uncertainties and the questions that we need answered before we can move intelligently and safely to the next phase of clinical development. So I haven't noticed any questions in the chat box. If you have them, please put them in, and I will try to break appropriately to answer them. What I'd like to do now is to switch the tone a little bit from what was essentially a menu of questions and goals of early phase work and give you an example of an actual phase 2 dose finding trial that's intended to specifically answer the question of what dose should be carried forward and whether or not a phase 3 trial should be conducted. A question that was asked whether predictive power conflicts with clinical equipoise in enrolling study participants. So say, for example, one is in the enviable position that the predictive power of a study is 50%. And believe it or not, 50% is a very good predictive power because it means that there's a 50% chance that you're going to have a positive trial that confirms a benefit. That level of assuredness that the treatment is effective is not adequate for it to be adopted into clinical practice. And therefore, without running the confirmatory trial, society will be deprived of access to that therapy for its patients. I have not heard of cases where trials with designed powers and predictive powers have led to concerns either at the IRB level or at other levels of ethical review regarding there no longer being equipoise. There is a very, very nice and brief, meaning 2-page article published in JAMA recently by Alex London like the capital of the UK, that talks about the term of equipoise. And the reason I digress here is because the question suggests that perhaps you're thinking of equipoise as an exact balance in people's belief regarding the probability that the drug works or it doesn't work, that there's true uncertainty that's exactly 50-50. Equipoise, in fact, is a much more general term, and the requirement for randomization is that there is sufficient uncertainty so that reasonable and competent clinicians might adopt different treatment approaches. So I urge everyone, actually, to download, and it should be free, the article by Dr. London, who's a philosopher at Carnegie Mellon, because it will give you a much deeper insight in 2 pages into the concept of equipoise and the resulting ethical issues in randomization than you're likely to get from other sources. And Sam, we might want to see if we can download that and put that in box for everybody. The next question is, could you please comment what states of early phase trials can be, I'm going to put on my reading glasses here, escaped when repurposing existing drugs for new indications? Okay, great question. So sometimes we choose to test an existing drug, for example, one that's approved for one disease state, in a new patient population with a different disease. Often the pharmacokinetic data can be largely used from the prior work, although we have to be concerned that patients with different ages or renal functions or liver functions may metabolize the drug differently. There is also evidence that the side effect profile of drugs often does depend substantially on the disease burden within the population, and so that has to be considered as well. But it is absolutely true that often the traditional phase 1 work can be skipped when repurposing an existing drug, but of course it depends on the details of the underlying data for that drug and the extent of difference between the original population in which the drug was approved and your target population for your new indication. The next question is, can you please give a concrete example of how heterogeneity of treatment effect complicates a trial in your experience? So, for example, we recently were working on the design of a clinical trial for a strategy for the treatment of patients with ischemic stroke, in which the patients were characterized at the time of presentation based on the volume of the core infarcted brain tissue, the amount of the penumbra or the tissue that was ischemic and at risk but not yet believed to be infarcted. And so the patients had 3 different measures of their disease state: their neurologic presentation, characterized by an NIH stroke scale, the overall size of the stroke area, consisting of both core and penumbra, and the fraction of that stroke volume that was core and therefore thought to be irreversible. And the assumption was, and the belief was, that the reperfusion strategy being investigated was likely to depend highly on the heterogeneity of the disease state. So we had to design a trial that explicitly considered the possibility that the treatment would be effective in patients with some stroke characteristics and either ineffective or even harmful in others. And we wanted a trial that identified in phase 2 and then allowed that to be used as a phase 3 result, the patient population in terms of stroke volume that benefited from the treatment and for whom the treatment ought to be recommended. So that was an example in which we knew that there were things measured at baseline that likely strongly impacted both the efficacy of the treatment being studied and its likely safety profile. So what I'm going to do now is talk a little bit about adaptive trials, which is an extension of what we will be teaching in many parts of the course, but the trial illustrates the concept that we want in phase 2 to identify a dose that ought to be carried forward, and we must explicitly, for the phase 2 trial to be most informative, address the question of whether or not a phase 3 trial ought to be run at all. This is not a neurologic example, but it's one that's in the public domain, and the design paper has been published, and that makes it convenient to talk about. So the clinical setting is patients with severe sepsis or septic shock. It's a phase 2 dose-finding study, so it's a learn phase study. And our goals are multiple, as I mentioned is often the case for early phase work. We want to identify the most promising dose for carrying forward into a phase 3 study. We want to determine whether or not we should do a phase 3 study at all. And we would like, within the phase 2 study, to enroll more patients to the doses that, as we gain information, turn out to look most beneficial, both so we get the most data on the dose of interest, which helps us with our safety concerns, and also treats the patients within the study as effectively as possible, if in fact the different doses have different levels of benefit. L-carnitine is believed to work through reducing multi-organ system failure. There is a score called the SOFA, or Sequential Organ Failure Assessment Score, that characterizes this. It is a measure both of disease severity at presentation and as a progression of disease. So you could think of it as like a neurologic outcome score. So the baseline SOFA is a key predictor of mortality. A big number is worse. And a reduction in SOFA over 48 hours is what we think will be the most sensitive measure of the benefit of L-carnitine, if in fact L-carnitine works through reducing multi-organ system failure, which is the presumed mechanism of action. So L-carnitine being able to reduce the SOFA score over 48 hours in a dose-dependent way would be a proof of concept for this treatment approach. But we know that in a confirmatory trial, a registration trial, 28-day all-cause mortality is the traditional phase 3 endpoint for sepsis studies. So in order to determine whether or not we ought to run a phase 3 study at all, we're going to need to have some idea about whether or not we believe L-carnitine in the chosen dose can actually reduce 28-day all-cause mortality. So what is the structure of the trial? We're going to have 2 outcome measures. One is the change in SOFA score from baseline over the first 48 hours, with the idea that if that goes down so the delta is negative, that's a good thing. but we're going to have our definitive outcome measure to be survival at 28 days, the same outcome that's used in a confirmatory trial, because we want this trial to tell us whether we should run a confirmatory trial at all. It's a dose-finding study, and it is placebo-controlled, so we have a placebo arm with 0 grams, 6, 12, and 18 grams. This is an amino acid. It's present in the diet. It really is grams and not milligrams. And we have a maximum sample size of 250 subjects, which was in fact limited by the funding structure at the NIH Institute to which this went. I will tell you that if we had designed this without the limitation in funding, we would have wanted to run a larger phase 2 trial. We're going to conduct interim analyses at initially after 40 subjects, then every 12. This is a feature in an adaptive trial design because we're trying to learn most efficiently from the data that are accumulating. The adaptive feature is that we are going to randomize patients not equally across all of the doses, but we are going to adjust the randomization ratios so that as evidence comes in that one dose is better than another dose, we preferentially randomize to the dose that appears to be best so that we force it to perform and see if it's really best. This improves the likely outcomes for patients within the trial, but we maintain a fixed randomization proportion to the control arm so that we always have enough control patients to be able to unambiguously test hypotheses regarding the presence or absence of a treatment effect. The trial may be stopped early for success or futility. If we have demonstrated that no doses are adequately effective, we can stop early for futility. If we identify a dose that has a high enough probability of success in phase 3, so that's a predictive power, then we can stop early and move on to phase 3. So now I'm going to show you the results of some simulations of this trial. So these are not actual data, so that you can see how it performs. So under the assumed situation where there's no benefit at all the doses work exactly the same as placebo, we don't expect there to be any change in SOFA relative to placebo, so that's the column of zeros under delta SOFA under no effect. And then the patient population is expected to have a 28-day all-cause mortality of 40%. We're also going to consider what I'll call a mild effect scenario, where the highest doses of L-carnitine have an effect of -1 for 12 grams and -2 for 18 grams on the change in SOFA, so that's an improvement. And those changes in SOFA are tied to decreases in 28-day all-cause mortality. This hypothesized benefit that's dose-dependent, although it's labeled mild, would in fact be a phenomenally large benefit in the setting of sepsis research. So how well does the trial perform? Under the null scenario, where there's no benefit at all, there is a 4.3% chance that the trial will falsely decide that not only is there a dose that works, but that it works well enough that a phase 3 trial ought to be performed. So that's a type 1 error. It's a mistake. On the other hand, the trial has a 43% chance of stopping early for futility, which is a correct decision. A 2.3 chance of stopping early for success, that's an error, but it's already captured in the type 1 error rate of 4.3%. And on average, it takes about 200 subjects for that trial to stop because it sometimes stops before it gets to 250, most often for futility. Under the hypothesized situation in which there is a marked benefit in both organ failure and the associated mortality, the probability that the trial concludes that this drug ought to be tested in a further confirmatory trial and picks a dose is 91%, so that's a power. It is not a predictive power, but a power in the traditional sense, because this 91% chance of a positive result is under the assumption that there is the effect shown in the first set of rows of the figure or table. There is a 1% chance it'll stop early for futility. That would be a type 2 error. There's a 68% approximately chance it will stop early for success and recommend further testing under this scenario. And the average number of patients that are enrolled is about 172. Most importantly, this design, and I haven't told you enough about the rules to know how it works, but this design is highly accurate in selecting the 18-gram dose as the most effective dose. One of the problems of many traditional dose-finding approaches is that if one simulates them through computer simulation, you find they're actually not very accurate in selecting the dose that is in fact the best dose. And that's something to spend some time thinking about if you're doing work that involves dose finding. So there are approaches that are accepted but still aren't very good. How well does this trial design treat patients within the trial itself? Under the scenario where there's no benefit, and therefore it doesn't matter to a patient whether or not they're enrolled to any particular arm, about 32% of the patients are assigned to the control arm. That's a necessary assignment so the trial can actually answer the questions of interest. And the rest of the patients are roughly allocated to the 3 arms, which doesn't help or hurt anybody because none of the arms do anything under this scenario. Under the scenario labeled mild effect, which as I mentioned would actually be a substantial effect, 31% of patients are ultimately enrolled to the control group on average. And again, that's necessary so that we can control for any changes in secular trends over time. However, of the patients who are enrolled to the active arm, 48% of all patients, and the vast majority of those on the active arms, are ultimately enrolled in the 18-gram arm, which is the most effective arm. So this trial design, which uses response-adaptive randomization responsive to change in SOFA, manages to get almost half the patients on the most effective arm, just like a one-to-one trial would do, except you also got to answer the questions of whether the 12-gram or the 6-gram was effective or the most effective. The goal of the trial was to pick a dose based on finding the minimum dose that was as effective as the maximum dose. In this particular scenario, that choice is the 18-gram dose. So the adaptive design has the advantage of allowing you to treat patients effectively within the trial if in fact the drug works, and to get a large fraction of the patients on the most effective dose even when you didn't know what dose that was going to be a priori. I should mention that this trial is funded by the NIH. It's supported by NIGMS. The principal investigator is Alan Jones, an emergency physician at the University of Mississippi, a good friend and a very productive colleague. And it is currently enrolling patients. I have not presented any actual data from the trial because the trial is ongoing. So, in conclusion, the goal of an early phase clinical trial is to answer the questions. First, should the treatment be further investigated? So if you're in phase 1, should you proceed to phase 2? If you're in phase 2, should you proceed to phase 3? It should answer the question about how any further trial should be designed, conducted, and analyzed. A good phase 2 trial tells you how to design your phase 3 trial, or tells you what uncertainties are remaining that must be addressed in your phase 3 design. You want to avoid being in the situation in which you're making guesses about doses or populations or outcome measures, but in fact your choices in those areas are informed by good, solid learn phase data. The design of an early phase trial must be tailored to allow for greater uncertainty regarding the safety and the efficacy of the agent in the population, greater uncertainty regarding how best to administer the treatment, dosing, timing, dose adjustment, and how to measure the effect of the treatment. And therefore, there is a need to address multiple trial goals, and that's an inherent characteristic of early phase work. I will stop there, and I was hoping to leave time for questions, and I'd be happy to go back to any of the concepts or answer any related questions that come up. You could, again, please use the chat box for questions. That would be great, and I'll give people a minute. There's a question, are there any examples where adaptive trial design should be avoided? Absolutely. Adaptive trial designs should be avoided first when the additional design effort that is required to do an adaptive design and verify its operating characteristics is simply not justified by the type of work being done. If the disease is relatively mild, you're doing a symptomatic treatment. The patients are not substantially burdened by being assigned to less effective arms. Then the additional work and complexity associated with an adaptive design is simply not warranted. So think of things, for example, symptomatic treatments for dermatologic conditions where patients can ultimately be switched over to the other treatment, often without harm from the prior treatment. So diseases in which the disease state or the symptoms are highly reversible are often cases in which the adaptive design may not be warranted. Another broad class of situations in which the adaptive design has absolutely no benefit is one in which the time to information is long and the time to enrollment is relatively short. So there's no new data coming in to learn from, so there's nothing to adapt to. A good example of this would be trials of prophylactic strategies or trials of disease-modifying agents where the disease has a very long course. For example, there are a number of degenerative neurologic diseases, as you all know, that progress over years. And if you are enrolling patients and you're going to need to follow them for years on a standard treatment, you're going to need to assign all the patients early on before there's any information coming in, and there is nothing to be gained from an adaptive design that's responsive to outcomes because there are no outcomes to respond to. There are lots of other examples where adaptive design has relatively little benefit. Another general class are adaptive design in terms of response adaptive randomization, where we vary randomization proportions, where there's only two arms. Sometimes that may have benefit from a patient point of view, but from a statistical point of view, you only will lose statistical efficiency when you deviate from balanced one-to-one randomization. The term adaptive is very broad. It also includes things like traditional group sequential trials. Obviously, those trials have tremendous value, and we have decades of experience with them, and they have benefit under a large number of situations. The next question. For a proof of concept 3-arm study testing 2 doses of a drug versus placebo, are there specific rules for sample size selection for the control placebo group? Again, it's a good question. So a traditional design that includes 2 doses versus control might use 1-to-1-to-1 balanced randomization and have that be fixed over time. If one is going to change the randomization ratio over time, one can do so with the goal of having greater number of patients on the dose that's ultimately of most interest. So for example, if your highest dose start to experience a high rate of dose-limiting toxicities, but you're not quite yet sure if that rate is too low to exclude the dose from further evaluation, you may want to have a greater fraction of your future patients on the lower dose, but still have some patients on the higher dose while you figure out with more accuracy what the dose efficacy and dose toxicity relationships look like. The reason you want to maintain patients on a control arm, even, for example, after a situation in which you think you are pretty sure that the active treatment is better, is because if there are secular trends over time, having patients on the control arm throughout the temporal duration of your study can be very helpful in understanding that, even if that temporal trend is not taken into account in your primary analysis. For example, you may want to do secondary analysis to show to what extent the date at which the patient was enrolled affected outcome. If you change your randomization ratios over time, so you say start to emphasize the lower dose, then you're going to have the lower dose arm is going to have more patients enrolled later than the higher dose arm, so understanding secular trends becomes particularly important. The next question is, are there examples of early phase maximum tolerated dose or CRM approaches in which adaptive design has been used? So the continual reassessment method is a type of early phase adaptive design that's very well established, in which the dose allocations for patients in each stage or step or group of the trial is continually updated based on prior data. So a CRM design is an adaptive design at its very core. And again, as I said, it is probably the de facto standard now, certainly within the for-profit pharmaceutical industry, for dose finding, because it's more efficient and frankly it's safer for the patients. Additional questions? So the question is, why would I have preferred a larger sample in the adaptive trial example you shared with us? Well, I made the comment that the middle size assumed treatment effect, which was labeled as mild, would have actually turned out, if it is verified as being true, as being a monumental improvement in the treatment of patients with sepsis. So we were forced to design a trial that had adequate power to detect a benefit only for benefits that were much larger than those that would have actually been of clinical interest. So ideally, we would like, especially in diseases that are very hard to find effective treatments for, sepsis, Alzheimer's disease, stroke, neurotrauma, we would like to design trials that are highly likely to detect any treatment that is treatment benefit that is large enough to be of clinical importance. When we design smaller trials, we're left in the uncomfortable situation of sometimes having results in which we are afraid the benefit existed, but we failed to demonstrate it, leaving it unclear whether or not the treatment ought to be investigated further. Whenever you read a report and the final result, the conclusion is, we were unable to demonstrate a benefit of X on Y, but it deserves further study, that comment to me is code for saying we had an inadequate trial design because we've left the question on the table even after completing our planned study. And the trial that I presented here could end up in the situation where there is a hint of benefit that is tantalizing and of a magnitude that could be clinically important if verified, but the trial is unable to determine whether or not it is likely enough to be real to warrant further testing. And in fact, I will say that the evaluation of the grant application from the NIH acknowledged that the trial was limited by funding availability and its size, and one of the reasons they funded the trial was not just enthusiasm over the treatment being investigated, but an enthusiasm over the trial design approach and what it illustrated in terms of the way NIH funded dose-finding work ought to be designed in the future. Are the terms proximate versus definitive outcomes only used in early phase trials? So I think those are in fact more general terms, and obviously we mean proximate in terms of temporal relationship. And even in a confirmatory phase trial, for example, picture a trial with a 6-month modified Rankin or neurologic outcome score. You're going to have more proximate measures of neurologic outcome, such as the outcome at 1 month, that in fact are highly informative. And some neurologic outcomes at 1 month, like death, in fact, are completely informative. So we use these terms generally in all sorts of clinical trial design work, and keeping clear in your mind whether your treatment is more likely to affect proximate outcomes but not long-term ones is very helpful in making sure we don't get fooled by signals that are limited to proximate or only temporary treatment effects. The last question I think I should take in the interest of time is, just slipped, hold on a second. And I will do the one that just slipped in. In adaptive design trials, do you ever get worried that due to the high number of treatment groups, the treatment groups may not be balanced on important confounders? We do worry about this when we simulate the trials. We look at the probability of getting an imbalance in confounders that is important in terms of, or concerning in terms of the conclusions that are drawn. I can tell you that our experience both in simulation work and the actual conduct of these trials is that people worry way too much about imbalance in confounders and worry way too little on things like the uncertainty of the patient population or dose or outcome measure going into the trial. And when we have more time together, including at the residential course, I'd be happy to talk about that in great detail. It's something of interest to me. The final question says, in behavioral interventions, the dose can be 4 weeks, 6 weeks, or 8-week therapy. Could you do a 1-to-1 8-week treatment versus placebo and test the outcomes at 4 weeks, 6 weeks as a way to address dose? So if, in fact, you have a long course of treatments and the 8-week treatment is the same as the 4-week except it's just twice as long, then you create a situation where during the first 4 weeks of treatment, the patient and the therapist has no idea whether they're going to stop at 4 weeks or 6 weeks or 8 weeks. Then there are some clever ways to use the repeated measures of outcome over time to determine the optimal, necessary, or minimally effective duration of treatment. This is a very long conversation, but there are absolutely things that could be done as long as what's done in the first week, the second week, third week, and fourth week is completely unaffected by knowledge of whether or not the treatment is going to stop in 4, 6, or 8 weeks. Another great question. I'd love to have the opportunity to talk with you more about that. I think that we are, in fact, over the hour, which means at some point Sam will start waving in her in the video box. I really appreciate everybody's time, and I really ask that you complete the evaluation form, not just for CME, but so that we can make these webinars as effective as possible. Thank you very much, and I look forward to meeting everybody at the course.
Thank you, Dr. Lewis, for that fantastic talk, and thank you everyone for attending. Please complete the evaluation. You can click Browse To in the web links box here. If you want CMEs, you'll be asked to provide an email address, and we'll provide the CMEs at the end of the webinar series in November. So our next webinar will be on May 12th at 12 p.m. Eastern. The topic will be Rigor and Transparency, how to use preclinical data to inform trials. And it will be presented by Dr. Lori Gutman and Dr. Will Moyer. So look forward to seeing everyone next time. Thank you.
Thank you, everybody.